Okay, so 15 nitrogen actually labels the base in the DNA strand, okay? So remember, nucleotide actually has base and phosphate groups, right? So this base is a nitrogen base, meaning this contains nitrogens, okay? So all of these nitrogens in the nucleotide is actually labeled with 15 nitrogen, okay? So that's the parent. And then after one generation, so when it goes through one duplication, this is put in a different environment. So all this environment actually has 14 nitrogen instead of 15 nitrogen, okay? So we provided this environment with 14 nitrogen only. Then what happens is these two actually get separated, 15N, okay? And then the new copy of DNA actually gets labeled with 14N, and then 14N like this, okay? And then if you do another duplication, then these two actually get separated, so 15N, 14N, and then 15N like this, okay? And then you keep that environment. So this is also carried out in 14N environment. Then whatever that gets copied is also 14N, all right? And this is 14N, okay? This is 14N, okay? This is also 14N, all right? So when you look at 4 double strands, okay, for DNA, then these two are 14 to 14, 14 pair. 14 pair, okay? And these two is actually 15 pair, okay? So as a result, when you actually figure out whatever that gets produced here, there are 2 types of DNA, okay? Which is 14 and then 15, all right? So that actually proves the model for semi-conservative, okay? That came up from the Messelson and Stahl, all right? All right, so if we understand the semi-conservative model, all right, then on page 135, let's study further on the structure of DNA, okay? So on that page, first of all, the first point, DNA is a double helix shaped like a twisted ladder, okay? And then DNA consists of 2 complementary strands running in opposite directions, okay? So when you go from 5 to 3, then the other one goes from 3 to 5 like this, okay? And these are these two, okay? These two are called complementary because the base is actually bonded to another base, okay, that is complementary to each other. So these bases are actually paired between purine and pyrimidine, all right? So purine, which is A, bonds to pyrimidine, and another purine binds to pyrimidine, all right? So when you have a sequence that has like A, C, G, T, A, okay? Then the other complementary strand, okay, actually has the base of T, G, C, A, T, okay? So whatever that follows, this complementary base pair, okay? And always you have to remember that hydrogen bonds between A and T is 2 hydrogen bonds, and this is 3 triple bonds of hydrogen bond between G and C. And then number 3, it is a polymer made of repeating units called nucleotides, okay? So as we looked at before, yeah, I rearranged, okay. So there were a series of nucleotides bonded to opposite ends, okay? So in the opposite direction, it has repeating units of nucleotides. And then each nucleotide consists of 5 carbon sugar, which is deoxyribose, right? Remember for DNA, okay, so this is deoxyribose, meaning the second, okay, so this is base, and this is C phosphates, right? Okay, so when you look at the nucleotide that makes up the DNA, then always the second carbon has hydrogen instead of OH, okay? So this sugar is called deoxyribose because it doesn't have oxygen at the second carbon, okay? If you consider this, if you compare this with the nucleotide for RNA, RNA actually has the same structure for nucleotide, but at the second carbon, it has OH instead of H, okay? So this is the difference.